Hello, I'm Dr. Mark Williamson. Before continuing on to the main video, please take a moment to take our pretest, which is found at the link below, on our webpage, or in the links to the description of this video. Also, when you're done with this presentation, please take our post-test and survey. Thank you and enjoy the video.
Greetings. Prepare yourself to run the statistical gauntlet using R. I'm Dr. Mark Williamson for the Biostatistics, Epidemiology, and Research Design Core for DaCCoTA at UND. Let's get into the purpose behind this presentation. Learning statistics is a tricky business. It takes time and effort. Now, unfortunately, a lot of introductory statistic courses, when introducing a new model or method, will only provide one or two examples. This makes this really hard if you have your own work, your own data you're trying to use, and you'd like to examine and sample to translate it for your work. It's hard to do that if you have a single sample, a single example with its own particularities and nuances. That doesn't really translate to yours very well. This use of only one or two examples also is hard when you're trying to train your eye to sort of get a feel for how to understand statistics, both from the standpoint of running the statistics and the standpoint of examining and interpreting the results. Because really what's often goes on with statistics is you have a rather concrete and plain research question that you have. Say you're interested in seeing if a certain drug reduces the risk of a certain disease. But to get that answer, you have to step into two rather abstract realms. The first is the statistical methods themselves, and the second is the computer and coding to run those statistical methods before you can spit out yet another concrete answer to that concrete question you had. And so it takes a lot of work to navigate those abstract fields. So a big part of this presentation is to remedy the issue of one or a few examples by providing what I would say it be much more exhaustive back-to-back examples, one after another, bam, 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 running the gauntlet, hence. And this should be helpful for both introductory and intermediate learners. Now, if you're pretty familiar with statistics, feel free to stick around. You might learn or refresh a thing or two. And I also have provided some code in a supplemental text that might have some interesting things that you don't have yourself. Let's turn now to a bit of an overview. So we'll be using R, just as our title suggested. So if you don't already have R software, you can get it via this website here. And I strongly would suggest also getting the R Studio software. It's a graphical user interface. It makes things a lot easier in the long run. And then all the code that we will walk through ultimately can be found here. And in fact, I've even annotated and provided a relatively clear format of the code, so it's not just snippets of otherwise incomprehensible gibberish that you have to wade your way through. It's not the most user-friendly, but it does have some semblances of friendliness to outside eyes. And all the data sets I'll be using today are available in R via various packages, the base set, or web link, and that's of course included in the text. And so we'll be covering three broad topics, 9 sort of tests overall, 3 different t-tests from 1 sample, 2 sample, and paired. We'll cover ANOVA, analysis of variance, one-way, two-way, and then a block/nested ANOVA. And then finally regression, starting with simple, on to multiple, and then finally logistic regression. Let's go over our procedures so you can appropriately brace yourself for what's about to befall you. I will be providing 6 examples per topic. So if you do the math, or let your trusty calculator do the math for you, that's 56 different samples. Each of them will include a test statistic, a p-value, and then where appropriate, model fit. And I have those outlined by color, these colored bars. And each example will kind of include the format. First of all, I'll state the research question in the form of a sentence, so to be as concrete and clear as I can. And I'll provide relevant statistical results from R. Now, I never promised you that R would be clean. It's actually rather messy, but it gets the job done. So I will clean up the results relevantly, rounding most values to 2 decimal places. However, I will not modify the p-values because I find it's just important to kind of leave them as is. Then I will provide a written answer to the research question, again trying to enunciate the results as clear as I can, and then I'll provide a figure or table when appropriate. Now, I'll have some graphs which I will consider for null results, and they'll be there in grayscale or red. These just include for clarity. You typically don't graph non-significant results. You don't put that in a publication, but for here, I think it's enlightening to do so. All right, with that all out of the way, get ready to run the gauntlet. Let's ease our way in here with 1 sample t-tests. These test if a variable's mean is different from a set value. It's one of the simplest tests you can do. So for example 1, it's the average miles per gallon of motor vehicles less than 25 for this 1975 data set. Can put this into R. Here's some of the results. First, we have our test statistic in the form of a t-value. It's important to note that test statistics can really all be boiled down to a signal to noise ratio. Our signal is if something significant is going on, and the noise is the variability that kind of gets in the way of obscuring that. So the larger our test statistic is, the more likely it is to be significant. And then typically, test statistics will have a p-value attached to it, which tells you the significance. Any p-values that are less than 0.05 will be significant. If they're above that, they're generally not considered significant. So what we have here, we have a relatively large t-value of -4.6 and a p-value. This is in scientific notation, so this is much less than 0.05. And indeed, we have our mean of our motor vehicles, and just eyeballing, we can see that, yeah, it's lower than 25, and that lowerness is significant. I also have confidence intervals here. Since this is less than, it's only one-sided, this would be a one-tailed test. We don't really need to look at the upper confidence limit, and that is less than 25. So now we can ask a similar question, but now asking if its quarter mile time in seconds is greater than 17.5. Test statistics, p-value, we see that now it's above 0.05, which means no. Quarter mile time is not greater than 70.5. The actual mean is a little bit higher, but it's not significant. We could ask a third question about these vehicles. Is the average weight different than 3? So now this is what we call a two-sided test. We're not caring about greater or less than, just is it different? So it could be more than 3, it could be less than 3. And indeed what we find here is, no, it's actually not different from 3. Mean of 3.22 is not. Let's move on to a different data set. Is the log baseline number of seizures for a placebo group of epileptics different than 0? T-value, p-value, not significant. Seizure number is not different from 0. Another data set here is the average yield of barley for 1931, greater than 100. So barley yields, this is a t-value that's significant. So yeah, yield is greater than 100, and in fact we can see the mean is 109, and that's significant. We could ask yield of barley for 1932. Is that different from, say, 1931's, which we know is 109? And again, that's significant. Yes, the yield is different than 109 because it's actually quite a bit less. That concludes our one sample t-test. Let's mosey on to a little more complicated version with the two sample t-test. It tests if the means of two different groups are different. So instead of having the mean of something compared to sort of a static value that we are interested in, we have two different groups and see if their mean is different. So let's go back to our good old-fashioned motor trans vehicles, and we ask if the MPG is different between types of engines, V-shaped versus straight engines. T-value is significant with our p-value, and we can see our means are quite a bit different. Straight engines have higher MPG than V-shaped ones, and we can actually do a nice graph here. We see that these straight engines have higher MPG. Let's look at quarter mile time, but this time between automatic and manual transmission. And turns out this is not significant. So this is a null result here. We wouldn't typically graph it. You can see here that the transmission type of 0, so that's automatic, does look like it has a little slower time or longer time, but it's not significantly different. Now, let's take a look at average tooth length of guinea pigs between a couple different vitamin C supplements, orange juice or ascorbic acid. Again, not significant. While it looks like OJ results in longer tooth length, it's not significantly different from our other group. What about average weight in chicks that are fed linseed versus sunflower? Well, that looks like it's significant, and we can see here that chicks that were fed linseed were lower than ones that were fed sunflower. So sunflower had higher mean weight. What about change in hours of sleep, so extra, different between two groups of students? So t-value, it's close, but it's not significant. So, and we can see here there's a lot of variation between the two, given these 95% confidence intervals. And finally, is the number of warp breaks per loom different between two types of wool? Again, not significant. While group A had its mean was higher, it wasn't significantly higher than group B. They do not have different number of warp breaks. Let's pivot to paired t-tests, which are similar to two sample t-tests, but in this case, the means of two different groups that are paired. They're related in some way, oftentimes a before and after sort of treatment. So first, we're going to revisit a data set we just used for the two sample t-tests, the change in sleep between two different groups. And now we learn, digging in with more information, that it's actually not groups, but the same student before one and after two a drug. And now we actually find that it's significant, taking into account the paired nature of the data. Students had a higher change in sleep after the drug compared to before. And I like to, you can use those bar charts like we did for the two sample, but for these I kind of like to use spaghetti plots just to show that sort of information between them. We see that students here before the drug, and then almost all of the bars go up. Sorry, the lines go up after the drug. Now let's ask about average weight of anorexic females. And now we're comparing post and pre-study period for females who are just in the control group. We find that that is not significant. weights were not higher in the post-study compared to the pre-study. And we can see it's kind of all over the place. It averages out. What about the same sort of thing for pre and post for a cognitive behavioral treatment group? So they're actually treating these women for their anorexia. And we find that it's significant. Female weights were higher in the post-study than the pre-study, which means on average it helped the anorexics gained weight, which was what they're hoping for. Now let's continue our anorexic data set, and let's look at yet another group, the family treatment group. So it's still a treatment group, it's a different type of treatment. And again, it is significant. We see here that not for every single female, it didn't work for every female, just like up here. Some females actually went down a little bit, but most of them tend to go up. All right, turning to 5. Is the average blink rate different between participants in steering a pencil along a straight or an oscillating track? So you can see a theme here that a lot of paired data is like the same individual measured at 2 different intervals or 2 different sort of scenarios. So again, we take a look. P-value is significant, and if you notice, I've been including these mean differences, and here in that sense of mean difference to be significant, it has to be significantly different from 1, far enough away from 1, given the data. And yeah, it turned out this is significant. Blink rates were higher in the straight track compared to the oscillating track. So blink rates were lower in oscillating, and pretty much everyone had higher, looks like everyone had higher blink rate across the straight path. And finally, is average blood lead level lower in control children compared to exposed children? So these weren't the same children, but they were sort of paired in a way, I think, to have one child be the same, more or less the same circumstances as another child, except for this exposure to, I think, lead in the water. And it looks like that was significant. The ones who are exposed tend to have higher blood lead levels compared to control. You can see that there's a fair amount of spread, however. Okay, take a breath, breathe, rub your bruises, and give yourself a second before we process into our ANOVAs. ANOVAs are testing, at least in a one-way ANOVA, if a variable's mean is different between a category with 3 or more groups. So this is really sort of the logical extension of a t-test, which looks at 2 different groups to more groups in that category. So not too much more difficult, but it'll start to need more nuance and intelligibility in examining the results. So let's look at that. See if the average sepal length is different across 3 species of iris, of these flowers. So now we're, instead of a t-value, we're going to be using f values. And we see that this is significant. And so unlike a t-test, we can sort of just look at the means. A significant result in ANOVA doesn't tell you which ones are different from each other. It tells you just at least one comparison is different. So typically you would do a post hoc test. I typically do Tukey's post hoc, honestly significant different test. So a lot of times you can get graphs like this, and it does every comparison. So one species against the second species against the third, second against the third. And if these are differences don't overlap one, they're significant. So it looks like all interactions are, or all comparisons are significant. And actually I'll plot that Ceratosa had the shortest, Virginica had the longest, and then Versicolor was in the middle but still distinct from them. And I just added these little asterisks to show that they were all significant. That would be one way of doing it. So now let's ask about the same 3 species, but let's look at, instead of length, sepal width. Again, that is significant. And look at comparisons, look like they're all significant. So again, we'll do the same sort of thing. It looks like this time Setosa had the widest, while Versicolor had the lowest, and then Virginica was in the middle. Let's look at something else here now. Different data set, but still in the plant realm, because it's easy to measure stuff on plants. Okay, give plant scientists a break. So is the average plant growth, so in weight, different across these different treatments? So we got F value, P value. Yep, it's significant. And then we look here, but only looks like this comparison is significant. So it looks like treatment 2 had higher weights than treatment 1. So I did a bar here to show that this interaction was significant, while this looks like this is the control. It was not different from either of the two of them. So that's another way to kind of show which comparisons were significant. Continuing with one-way ANOVA, let's look at question 4. Is the average highway miles per gallon different across 3 car manufacturers? So this is another car data set. Yeah, manufacturers. And we find that this is not significant, that miles per gallon is not different against the 3. And this is, again, the comparisons. And so this is across Chevrolet, Chrysler, and Dodge, and there's no difference in miles per gallon. So it doesn't matter which car you buy, you get the same MPG on average for these cars. Now, continue with this data set. Is average highway MPG different against 4 types of cars? So we'll look at that, and that is significant. So we're comparing, now we have 4 different groups, so there's more comparisons, getting more complicated, but it looks like this, and this comparison are significant, and we parse that out. So it turns out small cars had higher miles per gallon, not surprising, these ones, than all the other 3, compact, midsize, and sport. And now, since we have more groups, often what you'll see is you see letters denoting significance. So B is significantly different from A, but then all A's don't have a significant difference. Only these 3 are different from B. Now 6, let's turn to insects, because again, you can count insects pretty nicely, just like you can count things on plants. Is the average number of insects after spraying different across 6 types of sprays? So wow, you can see we did 3 groups, we did 4 groups, heck, we can do 6 groups. And hey, that's significant. And so there's a lot more comparisons, so I'm going to just skip right to our nice graph. And now we can see that spray types C, D, and E, they're all significantly lower than A, B, and F. And now you see I stopped using letters because our spray types were in letters, so I just use symbols just to not get things too mixed up. So that'd be another way to do things. Okay, so we saw in ANOVA we could have 3 or more groups for a one-way ANOVA. Now two-way ANOVA increases it in another direction, which is looking at 2 categories. And really, two-way ANOVA is a class in the class of ANOVAs called multi-way ANOVA, where you could have 2-way, 3-way, even 4-way ANOVA, though the more categories you stack, the more and more complicated the analysis and interpretation, more importantly, of the results become. So we'll just stick with 2-way for now. I mean, how many ANOVA gauntlets do you really want to come flying at you? So let's start with number 1, our old friend 1975 motor trends. We're going to look at engine and transmission type. We looked at them separately before, but let's look at them together. And now we're going to have to start parsing out our variables and seeing their significance separately. And we see that we have F values and P values, and it looks like on their own, engine type and transmission type are significant, but the interaction, this VS.am, that was not. Interactions are always interesting So we'll plot those as they're significant. If they're not, we'll just do simpler plots. So you can see here as miles per gallon was higher, cars with straight engines and manual transmission. Now let's look at average gross horsepower between these two categories and see, again here, interaction was not significant, but neither was the transmission type. Only engine type was significant, with the V-shaped engines having higher horsepower. So think the mighty V8. Go figure. All right, let's reassess our guinea pigs that we saw earlier, but we'll look at both supplement, what we saw before, and dose. So two different variables. Dose, we can see here, you can think of it as numerical, but you can also treat it as a category, which we're doing here. We find here that all three are variables: the supplement, the dose, and the interaction were significant. So let's plot the interaction. And what we see here is length certainly increased with increasing dosage regardless. And it definitely differed between orange juice and the ascorbic acid for things like the lowest dose. It looks like OJ was more tooth length compared to the ascorbic acid on the lowest dose, but then it sort of disappears more at the higher doses. So there we go. Let's keep going here in 2A. Stay strong, stay brave. Let's reassess warp breaks per loom by adding not only wool like we did before with our t-test, but tension. We find that while wool is not significant, tension and the interaction term is significant. So we'll plot that here. It looks like when it comes to breaks in your wool, the grouping of low tension and wool type A has the highest breaks. It's significantly higher than all the other categories. All right, let's turn to 5. Average yields for peas. Is it different across nitrogen, phosphorus applications? So these are going to be either nitrogen yes or no, phosphorus yes or no. And what we see here is that only nitrogen is significant. It was higher in nitrogen applications, so a 1. Otherwise, it was not significant. And 6, let's take a look at diamonds. The average price of diamonds different across cut and color, and this is a subset of this full data set. I just included not all categories because otherwise it would have been very messy. And what we found here is that, yeah, they're all significant, both cut, color, and the interaction. And really there's a lot going on here. Sorry, this is grayscale. It's meant to be significant. I just thought diamonds, kind of white and other colors. Anyways, but lots going on here with these interactions, but a good take home here is that the cut of premium diamond and color F had the highest price, whereas the ones with the lowest was like actually ideal diamonds with actually really good colors, D and E. So actually I think what's going on here, why would some of the nicest diamonds be the cheapest? It's probably because there's other things going on that we aren't modeling here, like say probably size, but you can see here that definitely color and cut do play an effect. Okay, let's get to the trickiest types of ANOVA you'll usually see, which are either block or alternately called nested ANOVA. So that the test of a variable is used different across categories, just like an ANOVA it is, while accounting or sort of correcting for either a blocking effect or a nesting effect. A good way to think of a blocking effect is usually, it's also used in other contexts as a random effect. You might think of you might do treatments on a bunch of different plots in like a field, and different parts of the field might have slightly different characteristics that you want to kind of control for, even though you're not interested in that. That helps mitigate some of the natural variation going on. And then nesting is, think of as like those like kind of Russian dolls, they nest inside, that one variable might be entirely contained within another variable, and we'll take a look at what that entails in our coming examples. So example 1 is average yield of oats per these different subplots different across 3 varieties of oats. Those are the thing we care about, while controlling for the block it comes from. So we get our variable, and now we don't really include our blocking variable in our variable list. You can do some comparisons to see whether blocking mattered or not, but we're not getting into that here. The important part is that this was not significant. So even after controlling for block, the average yield was not different across these different varieties. Victory was not different from Golden Rain, which was not different from Marvelous. Despite these great oat variety names, they were not different. Let's continue our plant theme. Is average yield for peas different across nitrogen and potassium while controlling for block? So similar to what we did before, only we're using potassium and then controlling for block. And what we see here is that nitrogen and potassium were significant from the interaction, even controlling for block. And so yields were higher in nitrogen, like we saw before, and lower in potassium. So let's continue on. Another sort of plot here is the decrease in honeybees different across spray type while controlling for row and column. So you can control for more than one thing. So we have 2 different blocks. We're blocking by row and by column. And so treatment, that was highly significant. And let's plot that. And yeah, it looks like the decrease in honeybees was lower in sprays A and B, so those were significantly different than E through H, which have a different symbol. And then these ones, which have one of each symbol, that means like they weren't different from these or different from here. They're kind of in between, but they're not. You can't simply separate them between either this group or this group. And you control here for row and column. Moving on here is log optimal density. So I think this is in like an assay, so microplates. Different cross samples when nested in block. So here it's a little confusing because they're using block in a way that implies nesting, not so much blocking. So a little confusing. But again, we're not going to show nesting because that's something we're trying to correct for. And we find that this is a little tricky here. So we ran this initially using a certain function, and it appeared to be significant. That's why I've shown here. But then we actually had to use it because we had to use a different function, LME. We actually didn't find it was different, even when nesting block. And so this is that sort of ANOVA style comparison, and you see that all of these intersect at 0. And then again, this is all these comparisons to all of these p-values were not significant. So while the overall model appeared to be significant using a simpler method, digging more into it shows that there really wasn't a difference. Okay, turning to 5 is the number of words recollected. So these are different, like people doing this, different across word type, which they name variance, while nested in subject. So each subject was tested on these 3 different word types. We find that that was significant. So what we're showing here is there is like negative words, neutral words, and positive words. And it looks like people recalled positive words at the highest rate, and then negative words, and then neutral words, which is unsurprising. Neutral words you forget easier than more strongly emotionally charged words. Now, to wrap up our block and nester ANOVA, let's repeat this same thing here, but now let's also have a category called task, which is also nested in subject. So task, variance, and their interaction, we find there that actually now none of it was significant, including that more complicated structure. All right, take another break. Breathe. Breathe. You're doing great. Don't let anyone tell you differently. You are a champion. We'll get through this regression. We'll get through this gauntlet. Stay with me here. Now for regression, we're going to switch gears. For t-tests and ANOVA, we had our y variable, which was numerical, and then our x variable or x variables were categories, like categorical variables. This time we're going to test the relationship between a numerical response variable, that's our y variable, and then one numerical predictor variable. So now our numerical variable in our predictor, it's, well, numerical. And we typically plot the results with scatter plots, so a little bit different. But conceptually, very similar to what we've been doing. We'll get test statistics and so forth. So without further ado, let's look at yet another car data set. Is there a relationship between car speed and stopping distance? So here, I'm going to show intercepts because that is something that's important for graphing, though I'll just focus on the variables that we're interested in. So distance had an estimate. That's going to be the slope of our line of relationship between these two variables. We, of course, have a t-value and a p-value, and so distance was significant, and the estimate was positive, so there is a positive relationship. So I'm plotting it here. Car speed increased with stopping distance, so the stopping distance, as that went up, so did the speed. So let's look at another relationship. Is there a relationship between the log of an animal's brain, weight of its brain, and his body? Is there a relationship? And yep, looks like that is significant. As body weight goes up, so does brain weight. Then is there a relationship between, now this is some geology, so bear with me. Is there a relationship between the area pore space, so it's just an area, and perimeter in petroleum rock samples? Well, it looks like that is significant. There is a positive relationship. Pore space in rocks increase as perimeter increase, so the higher the perimeter, the higher the pore space. All right, let's keep going here. Again, on the rocks, we like to rock around here. We're going to look at pore space and permeability, and that is significant. But now we can see here that our estimate and our resultant t-value is actually negative, which means it's significant, but it was a negative relationship. You can have those as well. Pore space in rocks decreased as permeability increased. So you can have both positive and negative relationships. What's not significant is essentially a flat line, no relationship. But I'm getting ahead of myself. Let's turn to this Swiss province data set. Is there a relation between fertility rates and the percent of education beyond primary school? So education, find that there is a significance, and again, there is a negative relationship. The higher the percent education, the lower the fertility rate. And again, on that Swissy data set, we looked at agriculture and infant mortality, and there was no significant difference. That our estimate was very, very close to 1. T-value and p-value was not significant, and I just plot the no results. You can see essentially as agriculture changes, really infant mortality isn't really affected by it. So again, I'm using the word relationship because I think that is a helpful term when we look at simple linear regression. I'm not saying one causes the other because correlation does, of course, not imply causation. Every single regression example or textbook will tell you that, and I would be remiss if I didn't throw my weight behind that assertion as well. Okay, so multiple linear regression. Here we can have multiple numerical predictor variables. Again, sort of the logical extension. If you can have multi-way ANOVA, why not have multi-way linear regression? Why not? Yeah, exactly. So let's do it. So let's turn back to our rock samples and let's throw in perimeter and permeability to try to predict area at the same time. And now multiple linear regression starts to fall into the problem of what we call model fit, because a lot of times the more variables you add, and not all of them will matter or might interfere with each other, so you often want to do things to find the best model, which might involve simplifying. So we're going to start by looking at our perimeter and permeability, and it looks like both are significant. But then what we're going to do here is actually compare models using what's called AIC scores. And it turns out that the full model has the best fit. So lower means better fit. So just you have to get used to that. And so we could say that pore space increases, perimeter and permeability increased. Now we're not going to plot this because this starts to get into multiple dimensions, more than 2 dimensions, which is difficult to interpret. In fact, if you get more than, more than 2 numerical predictor variables, you can't plot it. So just bear with me here. Okay. 2, can frontal lobe size in crabs be predicted by rear width and body depth? So we're going to do this here. And I'm only showing body depth because what I ended up doing is looking at kind of simpler models of just the rear width only, the body depth only, or the stuff together. And while it turned out that this model had the best fit, it turns out an important thing when you have multiple predictor variables is if they're highly correlated, you shouldn't include them together. Kind of the hand-waving way to explain this is if they're very highly correlated, they're pretty much tell you the same thing, which is not useful. So what I did here is rear width and body width, they were highly correlated. I have this plot here. You can see pretty strongly correlated. I only took the single variable model, so this BD only, and I retained that. And it turns out, yeah, frontal lobe size increased as body depth increased. So again, let's look at these crabs again. If we hadn't learned our lesson already, look at carapace length and width. Again, what we did here is, this is the final stuff I get out here. But what I had done before is I looked, and again, these 2 variables were highly correlated, so I picked the single variable model, so that's the length one that had the best fit. And so it looks like frontal lobe size increased as carapace length increased. Okay, moving right along to more examples. Okay, we're gonna again turn to the Swiss data set and see if we can predict infant mortality rates by a bunch of different variables. Fertility, percent Catholic, percent of males in agriculture, and percent education. And so I'm going to show you a little bit different here. I'm going to show you the full model with everything in it, in which we see that fertility was only significant variable. And I also show you the simple model with just fertility itself. You can see that it's again significant. And the fit between the 2 models, it looks like the simple model is a little bit better fit, but whether or not that's low enough to use. So you could either use the full model to kind of show that these other variables that you might think would be important aren't, just to show that result, or you could use a simpler model. Either way, what we get here is that as fertility rate increased, so did infant mortality. Okay. Let's look at another example. Can calories per portion in U.S. cereals be predicted by protein, fat, carbohydrate, or their interaction? And this is going to be square root transform data, just to make it a little easier. So now I'm going to be showcasing a technique to find the best model called model selection, where you actually take the model, you run a certain procedure, and it'll tell you which variables you should retain. And so I did the full model here, and it had an AIC of this, And then this is the final model, which has a lower AIC. And so this one had all three variables and all the interactions. The final model had all three variables individually and then just the interaction between protein and carbohydrates. So with that final model, I ran my regression, and these are the t-values and the p-values we got. And I checked for correlation covariates. And so what we found here is that protein, fat, and then the interaction between protein and carbohydrates were significant as protein increased, calories decreased, while for fat and the interaction between these two, as those increased, so did calories. So let's wrap up here with good old cars. Can miles per gallon be predicted by gross horsepower, weight, and quarter-mile times, or their interactions? So again, we'll do model selections, where I start with all interactions. So that's what the star is the interaction between these two, this, and then all three-way interactions too. And the reduced model that it gave me was horsepower alone, weight alone, quarter time second alone, and then interaction between horsepower and weight to have a significantly lower AIC. And then, but that wasn't the final model I ended up using, because it turns out that horsepower was highly correlated with weight, so we only want to include either weight or horsepower. So that's why I compared these models, it looks like weight was had higher correlation, so I just included weight, and then of course the core second time, which ended up those two being significant, which means as weight increases, the miles per gallon decreases, not surprising, and as quarter second time increases, miles per gallon increases as well. So heavier cars have lower miles per gallon, and speedier off the line cars have higher miles per gallon. Okay, we've made it to our last regressions, logistic regression. So up until now, one of the assumptions for all these tests that I glossed over, in fact not even glossed, ignored, because I was trying to focus on something else here, is the assumption of normality, that your data comes, your y data, your y variable, comes from a normal distribution. It turns out a lot of data that's not the case, and so you can do other forms of tests. One such for regression is what's called logistic regression. This is when your response variable is binary or categorical, but we'll just look at binary data. So this would be the idea of, what if your response variable is something like disease status, yes, no, or it's alive status, alive, dead, something binary, 1, 0, yes, no, etc. So let's take a look at how you do this. So let's ask the question first. Can Midwest County's metropolitan status, so either yes or no, be predicted by the log population density? So we'll sort of spit these out. Now, instead of a F value or T value, we have another test statistic called a Z value, but giving us the same sort of thing. This is significant, so population density is significantly predicted. And what we plot here is a little bit different, because we actually have, since it's binary, we have to essentially plot a probability, and it often follows this sort of S-shaped curve because it's bounded by 1 and 0. So this is probability. So as log population density increases, the probability of being a metropolitan status increases, which should make sense. If a really dense county, it's almost certainly going to be metropolitan, and vice versa. Very sparse ones are going to be certainly non. So let's continue with this Midwest. can we detect status by percentage of college-educated adults? Maybe something a little bit less obvious. And turns out that is significant. And again, the more percentage of college-educated adults, the more likely it is to be metropolitan. Sort of makes sense. See here, it's not quite as strong of a curve, but we can still see that sort of S-shaped curve. Let's turn to one of our car data sets. Can we predict a car's origin, so USA versus non-USA? Can we predict it by city miles per gallon? And we find, and that this is significant, but it's an inverse relationship. So it turns out as city miles per gallon increases, the probability that a car's USA origin decreases. So foreign-made cars tend to have better mileage. So there we go, and we can see here that it's a very, very weak S-shaped curve. It's almost non-existent, but it's still sort of there. And we see that there's no perfect predictability. Even though the cars with the lowest miles per gallon only have about 75% chance of being a US car. So again, these are probabilities that we're plotting with these graphs. So taking an example again with car origin, can we do this by price? And we find here our z-value and a p-value that's not significant. And so it did not significantly predict origin status. And I just showed these in red. You can see here that there's really very little difference. You can't really predict it. Okay, let's turn to another data set. Can the presence of a bacteria species, so yes or no, that's our binary option, in children be predicted by drug status and compliance? And now we see here we're doing something a little different here. We're doing something that looks awful lot like ANOVA, because it kind of is, but it's also regression, because regression and ANOVA are sort of the same thing, using just different sort of variables, and conventionally we use these different terms, but really kind of the same underlying, under the hood process. So this is another pretty common kind of logistic regression where you have categorical variables. So we can look at this, plot the same sort of results. So now this is where R gets really messy. So we have z-values and p-values, but what is this actually telling us? So let's start with this first variable. This is SP. So that's the drug status. And then P stands for placebo, because that's what it was called. So this is saying the placebo, and then what's kind of not said here, as compared to the other one, is significant. Same thing with here, the compliance status. And this is low compliance status. That's significant compared to the high status. So really what is more useful that along with this is what's called odd ratios that kind of tell us what's going on here a little better. So I have the odds ratios here. So we have our two different categorical variables, and then this is the odds ratios along with confidence intervals. And so what this says here is that drug status, children with the placebo compared to those without, to the drug, were over twice as likely to have the bacteria compared to those, with the drug. So that's so an odds ratio of 1 would mean there's absolutely no difference between our comparison. So between the placebo and the drug, and then we can tell if it's significant whether or not this overlaps 1. So this is higher than 1. So what it's saying here is that twice as likely, and it's significant. Where we see here the high-low, this was lower than 1, so presumably that means the one with low compliance have lower odds of having the bacteria, but that's not significant because it overlaps 1. So let's take a look at this. Again, it takes a while to really piece these out, so hopefully this is a good start. So let's look at another question. Can the presence of a tuner, so tuner versus none, be predicted by sex and treatment. So here we got here, and looks like both treatment and sex are significant. So it looks like treatment and sex predicted the presence of tumors in rats, and rats given the treatment were over twice as likely, we can see this is over 2, as likely to have a tumor compared to the control. So this is essentially a tumor-causing drug. Even more interestingly, that male mice were way less likely. This is like over like 10 times less likely to have a tumor compared to females. You see that is another example of odds ratios, and it's very common to see in, like, say, epidemiological studies to have a bunch of different odds ratios, and you can do this for category variables with more than 2 categories. It just means there's more comparisons. So there we go. Oof. All right, you've made it through the gauntlet. I award you for your bravery. Like, go out and get like a cappuccino or something. You deserve it, okay? You deserve it. So before we finalize this, let's do a couple acknowledgements. The title image, this is from WikiCommons, so just to give reference to that classical painting here. And then examples 5 and 6 from Block next to ANOVA. This is based on this this example from this project here, and that website and code is also referenced in my R code itself, so you know where that came from. So then finally, this is part of DaCCoTA, so we're supported by a grant, so please reference us if you find us any of our work helpful in your research project. So thank you for your time. Go ice those bruises from that gauntlet, and I will see you in another presentation. Goodbye.